Hey gang, welcome to the first class that's really going to put together most of everything that we've been talking about thus far, right? So this is going to be the class that puts together a lot of the circulatory physiology and the way that it works and why it works. Going to be talking about pulmonary physiology a little bit, talking about how we're breathing, why we're breathing, and we're going to be also talking a bit about enzymes, energy systems, all that type of stuff. So realistically, starting today and really moving on through the rest of the semester, it's going to be comprehensive, and it's going to be assuming that you know what is going on. If you start getting lost on some of this, go back to some of the previous lectures. You're always more than welcome to reach out to me. I'm always available for you guys. You know that. So with that being said, go ahead and get started on acid-base balance. For those of you guys that have had chemistry already, this is probably going to be a little bit easy, but what I want to do is I want to remind you what exactly acid is, what exactly an acidic environment is, what exactly an alkaline environment is. And it's going to be important for a lot of our discussions. Before we can really start talking about that, we got to talk about ions. So what is an anion and what is a cation? Anions are going to have gained an electron. That prefix "an" is usually a negative. It's usually meaning something negative. So here we're talking about a negative ion. So we've actually gained an electron. Cations are going to be things that have actually lost electrons. The individual things, the substances that are actually going to be losing hydrogens or committing hydrogens into the environment or releasing hydrogen ions into the fluid or into the body are actually going to be considered acids. So any substance that releases hydrogen ions is going to be an acid. It could be a weak acid, it could be a very strong acid. Any substance that is inside of the body that's going to be consuming hydrogens or bringing hydrogens out of a solution or out of the tissue and putting it onto itself and overall reducing the concentration of these free hydrogen ions that are floating around is going to be considered a base, or it's going to be more alkaline. Concentration of hydrogen ions is a super important topic. And one of the ways that you're going to see me talking about or see me expressing hydrogen ion concentration is going to be a bracket H plus closed bracket. So that means anytime you see those brackets around an ion or those brackets around a substrate, around an enzyme or whatever, it tends to mean concentration of. So in this situation, it's going to mean concentration of hydrogen ions. So pH is essentially going to be the concentration of hydrogen ions. You can do some really goofy I could give you guys a whole bunch of goofy information on what exactly is pH with the log and how you log something and basically how you do calculus to figure out what the pH of something is. It's way too complicated. And realistically, what you guys need to know is that as the pH goes down, the hydrogen ion concentration is going up. So the more hydrogen ions that are accumulating, the more acidic the environment, the lower the pH, right? Vice versa, the less hydrogen ions, the more that we're removing hydrogen ions from an environment, the lower the hydrogen ion concentration, the higher the pH. Why do we care about acid-base balance? What is it inside of the body? Well, I could give you the easy answer. The easy answer would be like homeostasis, right? Like imagine SpongeBob SquarePants doing like the thing, right? Like homeostasis. Essentially, our body likes a very tight window of control. Anything that puts us out of that window of control is a deviation away from homeostasis, and our body always wants to find a way to return back to homeostasis. Acid load's no different. The acid, the level of acidity or the level of alkalinity, I hate that word, is going to essentially decide whether or not we're put into or taken out of homeostasis. So if we start to drop the pH, if we start to become more acidic, our body's going to fight to return back to a constant level. Essentially, what we are going to try to do is buffer the acid load that is generated as we exercise. Remember, the class is exercise physiology. We're trying to study the deviations of homeostasis, deviations away from homeostasis, that result as a cause, or that result from exercise, or result from exercise-based stress. So it's simply that. The reason we care about acid-base balance is because we're creating an acid load in the body with exercise. We got to figure out how we're going to get rid of that acid load. So 2 slides ago, I kind of threw in this light little concept of we can have strong acids or we can have weak acids. We can have strong bases or we can have weak bases. Essentially, all that I'm trying to get at here is that the more readily available, or the more readily a substance releases its hydrogen ions, the more likely it is to be a strong acid. The more likely something is to grab onto hydrogen ions and pull it in are going to be strong bases. So the level at which it's willing to dissociate or gain hydrogen ions is going to decide whether or not it's a strong acid or a strong base, or a weak acid or a weak base. If we're talking strong acids, a great example, sorry I couldn't talk there, is going to be sulfuric acid. So it's produced from metabolism of sulfur-containing amino acids. Don't need to know that. Basically, if you put it into water, it liberates all of its hydrogen ions. An example of a strong base is going to be bicarbonate, right? We've talked about bicarbonate before. You should be familiar with this, at least the term bicarbonate, because we talked about it when we were talking about, wait for it, CO2 transport, right? We've already covered this topic. It's not going to change. We're just going to get more in depth. So essentially what's going on with bicarbonate, and we'll talk about it in the next lecture, is that we're able to combine with hydrogen ions that are going to be released from the working muscle. And basically, the bicarbonate system is used to help buffer the level of acidity inside the body. So it's used to help bring us back to a level of homeostasis when we get put out of homeostasis as a result of exercise. Remember, that's why we care about this. So what does pH in the body look like? What does the acid load in the body look like? Well, in generally speaking, if you will, normal pH for arterial blood is going to be about 7.4, right? So we are not going to move from that 7.4 very often. It's actually pretty rare for us to be budged too far away from that. And typically, if we get budged too far away from that, it's not a good thing. Right? So we want to maintain that 7.4 as tightly as we can. Some people it's going to be a little bit higher. Some people it's going to be a little bit lower. In general, it's going to be about the same. As we decrease in pH, as we move towards this level of acidosis, we're going to be increasing the hydrogen ion accumulation. I've said this a lot because this is a very important topic for you to understand. It's a very important point for you to understand. And that means it's probably going to show up on a test, right? So what happens if our body becomes acidotic? Well, we start to experience largely global dysfunction. If we have a pH that changes too much outside of homeostasis, we start to have enzyme catastrophe, if you will. We start to denature enzymes, we start to break down enzymes, and when those enzymes become denatured or we change the shape of those enzymes, then we're going to lose function. We can change the shape of things like ATPase. If that happens, are we going to be able to have muscular contraction? No, right? If we have issues with stuff like hemoglobin, are we going to be able to bind oxygen? No, right? All of these things become super important because if we denature these enzymes with acid or with an increase in hydrogen ion concentration, we're going to essentially stop the body from working as we need it to. That's not good. On the opposite side, we can also have metabolic alkalosis, ALK, right? So acidosis and alkalosis. So as we become more basic, we can have a state of alkalosis develop. This is actually can sometimes occur when we start to hyperventilate. So we can actually force ourselves into a basic environment. And I'm not just talking about you that's drinking those frappuccinos or went and stood in line for pumpkin spice lattes. I see you, Carter Grant. In general, whichever direction we go, it's a deviation away from homeostasis. But the major take-home point is that our body likes to stay within a tight range. Any deviation away from that is going to affect enzyme function. We affect that enzyme function. We affect the ability of our body to work like it should. We affect our response to exercise. We affect our ability to perform exercise. If hydrogen ions are so important inside the body, how are we producing them? Where are they coming from? Why can't we just not produce hydrogen ions and the world is happy, right? Like it's a better place. Well, the actual primary cause of hydrogen ion production is relatively a hotly debated topic, if you will. And that's why I got Hulk Hogan. Some of you guys might not know who Hulk Hogan is. I've actually am old enough that I know who Hulk Hogan is. And then he actually had a daughter that tried to do singing because he was so famous. It was an absolute disaster. But regardless, welcome to the Thunderdome of exercise physiology. Essentially what this all boils down to, and why we have such a hot debate going on here, is that there's three essentially valid places, or there's three valid rationales behind where the predominance of hydrogen production is coming from when it comes to exercise. Regardless, it doesn't matter. We can get entrenched into 1, 2, or 3. We can get entrenched into option A, option B, or option C. I don't really care, right? What I care about is that there's a hydrogen ion production occurring, and our body has to get rid of that hydrogen ion production. However, we need to be able to discuss those three avenues. We need to be able to discuss in detail where these three hydrogen ion production sites are occurring, because they are all occurring. It's just which one is the predominant one that is up for debate. Essentially, one place that it could be coming from, in no particular order, because I don't want to offend anyone, exercise is going to be inducing the production of carbon dioxide. When carbon dioxide meets water, when it hits the fluid of the blood, it's going to produce carbonic acid, right? One level. 2, exercise-induced production of lactate. If you guys remember, if you go all the way back to glycolysis, and when we're talking about these energetic systems and the bioenergetic pathways, you kind of have two ways that you can go with lactate, or my fault. You have two ways that you can go with pyruvate, right? You can go up, get that wonderful hand stamp to get into Club Crabs, or you can produce lactate. When you produce lactate, there's also a hydrogen ion involved there. There's a release of a hydrogen ion, right? There's an NADH that comes into play here. Remember, this is where we're talking about NADH, NAD ratios. And then the third place is going to actually be exercise-induced ATP breakdown. So as we're breaking down ATP with the ATPase, inside of the muscle, we're breaking those myosin-actin chains, or we're breaking those myosin and actin bonds. We're actually going to be liberating a hydrogen ion at the same time. Will, I don't know why you didn't have this on your exam. Disappointed in you. You had everything else. So let's talk more in depth, right? What is the origin of the hydrogen ion if we're talking carbonic acid and we're talking carbon dioxide? Essentially, we have a carbon dioxide that's being produced from the oxidation of carbohydrates, fats, or proteins. Where is this coming into play? It's coming into play all the way at the top of the Krebs cycle. You remember this, right? As we're going from pyruvate to acetyl-CoA, we're going from 3 to 2 carbons, we lose a CO2, right? Like we're producing a CO2. That's where your carbon dioxide is coming from. As this carbon dioxide that gets produced during the transition step in Krebs cycle, we're going to be combining it with water in the environment, and we're going to be forming carbonic acid. Carbonic acid, as the name would suggest, is actually able to readily dissociate hydrogen ions, making it an acid. Something that's really interesting about carbon dioxide, outside of being considered a weak acid, is the fact that it is volatile. And what volatile acids are able to do, or what volatile substances in general are able to do, is it means that they're gas. And since they're a gas, we can actually exhale them, right? So right off the bat, we know that we are dealing with a volatile gas. It's also considered an acid, and we know that we can breathe it out. Why do we breathe? Remember, we talked about this when we were talking about respiratory physiology. The large reason why we exhale is to reduce CO2. You should be able to start logically putting together how we're handling some of the acid load that we're producing with exercise. As you guys might guess, as we're increasing in exercise intensity, or we're increasing in exercise duration, we're increasing in reliance on certain types of bioenergetic pathways where we're producing carbon dioxide, we're going to be producing greater levels of acid, and we're going to have to buffer those somehow. Right? Expiration. That's how. Area number 2 in which we could be producing hydrogen ions, or we could be producing this acid, is going to be in the production of lactate. right? So we talked about this. I'm not going to go back over glycolysis. I'm not going to be going back over pyruvate dehydrogenase or lactate dehydrogenase. You should already know this. If you don't, go back. But essentially, it is going to be a contributor to the hydrogen ion production, right? How we develop lactate is going to be a contributor to hydrogen ion production. Another place that we're going to be producing hydrogen ions is going to be from the breakdown of ATP using ATPase. So if you look over here on the right-hand side, we're going to have ATP, right? Adenosine triphosphate, 1-phosphate, 2-phosphate, 3-phosphate. If you look at the equation, you can see that as we break ATP down in the presence of water with ATPase, we form an ADP, adenosine diphosphate, an inorganic phosphate, and we also have liberated a hydrogen ion. We didn't talk about this in class before because I wanted you guys to understand everything this way, right? The PI, the inorganic phosphate, the ADP, and the ATP. Now we're just adding in one extra product, and that's a hydrogen ion. So we're actually producing a hydrogen ion. We're producing an acid load by breaking down ATP. How many times do we break down ATP to go through one skeletal muscle contraction? A ton, right? So you can see why we're able to produce a ton of acid that we have to eliminate during exercise. This is a pretty good summative figure for you guys, right? In the middle, we've got hydrogen ions. We've got an increasing hydrogen ion concentration. There's 3 primary sources for that. We're going to have carbonic acid, CO2. We're going to have lactate, and we're going to have ATP breakdown. Carbonic acid, CO2, production in the intermediate step going from pyruvate to acetyl-CoA, lactate in the formation of lactate going from pyruvate to lactate, and then as well as the ATPase in the breakdown of ATP to form ADP and PI. We also get that little last hydrogen. 3 primary ways that we're going to be making hydrogen ions here. This is going to be why we care, right? Let's take that why we care one step a little bit further. If we have high intensity exercise and we produce those hydrogen ions from one of those 3 phases, I don't care where it's coming from, pick one, it'll be coming from there. Pick all 3, it's coming from all 3. We're going to have negative enzymatic function. So, summary, 3 different functions, 3 different ways that we're creating hydrogen ions. Large hydrogen ion concentration, bad. It's going to denature enzymes. We can also inhibit the ability to produce ATP because we're breaking down or we're denaturing those enzymes. Something else that's really interesting is we can also have hydrogen competition for calcium binding sites on troponin. If that happens, what do you think is going to happen to what do you think is going to happen to muscle contraction? We're going to seize it up, right? Like if we're blocking calcium from binding to troponin, we're not going to be able to have that conformational shift to tropomyosin, not going to be able to get actin-myosin bonds or strong binding states. None of that if we have high levels of hydrogen. Could be a mechanism for fatigue. What do you think? Next video, we're going to be talking about how exactly we handle that acid load.